gentlemen, executives, members of AHPI, and all friends from the healthcare fraternity, a very good evening to you, a very good afternoon and good morning as well. An honor to welcome you all to this Member Connect program, a live webinar for an intellectual conversation with a very sought after celebrity guest, Dr. Gagandeep Kang from the healthcare industry and especially the vaccine industry. Sure, all of you will be eagerly waiting for this presentation. As duty commands, I'm going to steal exactly 2 minutes of the session time to update you all on our good work that's happening from AHPI. Request you to share the slide, please. AHPI is a pan-India association of healthcare providers, bridging the concerns of doctors, hospital owners, healthcare workers, and the public with the Government of India, thereby synergizing the efforts to achieve greater good together. AHPI is primarily run by the hospital owners and decision makers, decision providers across the country and has a strong past that's very positive in helping the times of need. Whether it is the Tamil Nadu flood or the emergency responses that were required, AHPI has always stood up to a mark. AHPI conferences are world famous and they've always been attended well by very good speakers as well as audiences. The legal capability and the success of AHPI is very clearly seen when the government interventions are very successful, especially with construction of ramps and Minimal Wages Act from the labor, and of course, the vaccination, all the hospitals joining together to give vaccination to the country. In all these issues, AHPI has had a very, very strong positive influence. AHPI has made recommendations to the Government of India through a letter to the Honorable Prime Minister to make vaccines available to all age groups above 18 with immediate effect from May 1st, 2021. AHPI is an integral part of the National Working Group formed to evolve measures and provide suggestions to the Government of India to solve this COVID crisis. Next slide, please.
நீங்க பிரசன்டரை பாரு வந்து எப்படி இருக்காங்க பாரு ஸ்டைலிஷ்
Valuable suggestions
வெள்ளூர்ல
mobilization of manpower across the country, networking the availability of oxygen cylinders according to rising demand, forming an oxygen committee to oversee the distribution of oxygen cylinders and supplies across the country. Addressing the concerns of medical students, AHPI has requested the government to allot additional 30 marks to the PG NEET aspirants this year who have worked in the COVID ICUs relentlessly since last few months. I'm sure this will be really welcome by the students and especially the postgraduates. AHPI always stood up against violence meted out to doctors. Even in the most recent incident that happened in Delhi, AHPI has made interventions very promptly in support of the healthcare professionals. Next, please. This is a very important slide according to me because all the healthcare providers in India charge very differently from region to region. And the costing of medical procedures is a very, very important topic of concern. Today, along with the cost accountants of India, AHPI has joined together to provide a universal costing of medical procedures all across in every hospital, in every region. And this is wonderful data that's been collected. And the government bodies were also involved in this study. This, I would say, is a landmark study by AHPI. Next, please. Whether it is research or any academic area, AHPI has provided the best of the best. Whether it is combining itself with ISRO or is it a Lancet Commission, these studies are all landmark studies which will be providing the best of the best academic products in future. Next, please. And in the legal interventions, AHPI Tamil Nadu chapter has done a lot of good work and still is continuing the good work. We are negotiating with the CGHS, trying to get the best rates possible for all the insurance patients. The revision of COVID rates, which have been allotted by the government, this is a very important area again, where a lot of cost is being incurred by the private hospitals, and the COVID rates that have been allotted by the government is far, far less than what is required. And one more area where AHPI is intervening and trying to make this better is trying to reduce the electricity rates of Tamil Nadu. We are looking forward to these landmark judgments and landmark changes in the near future. Next, please. These are some of the very interesting books published by AHPI in combination with the doctors and with the writers, with the authors. And these are all wonderful books to be placed and during work, very useful as well. Next, please. Any organization committed to quality will always give good courses and excellent training programs to manpower development. So these are the areas where we have developed very interesting courses which all healthcare institutions can take on and will benefit very surely from these. So thank you so much. This is a very small but truthful presentation as far as AHPI and its wonderful activities are concerned. I request you to take me back to the main window where we can go ahead and start with the presentation. Ever since our fight against COVID-19 began, we have been looking forward to vaccines. Now that they are valuable and available to use, we are clouded with questions on their timings, their availability, its related myths and variants. It makes us wonder whether vaccines are productive enough in their mitigatory efforts, or is it being counterproductive in the minds of the common man today? Here we have amidst us the woman who has earned her own rightful place in the vaccine industry and is ruling as the pride of our country. To introduce our special guest, let's call upon our moderator, Mr. SAMEER Mehta, the president of AHPI Tamil Nadu chapter, a good friend of mine, a medical socialite, a man of the masses in the business circles, and is the CEO of one of the most oldest yet the state-of-the-art hospitals in Chennai, the Mehta hospitals. So over to you, Sam. And before I hand over the session to you to introduce the speaker and take it forward, a small request from my side to the viewers to please do not wait till the end of the session to type in your questions to the speaker. We shall pull in the questions from the start of the program to save time towards the end of the session and hear a little more from our star speaker. I wish to express our special thanks to our sponsors of this event, ANI, CII, and the Society for Emergency Medicine, India. Without your valuable support, this would not have been possible. Now over to you, Sam. Thank you very much.
Thanks, Gayatri. So I think welcome. There's a lot of folks here. There's a lot of folks I understand on the Facebook Live pages. I'm getting the messages now. So guys, keep joining because the more you learn, the more it's useful. So I have two other requests. If you could mute yourselves, please, other than Dr. GAGANDEEP and at times me, it would be. That's one. And Shiva, if you can, please, or admin, if you can mute everybody else, that would be useful. That is the expectation of the role I have for admin checking. The second point is, please type in your questions in the chat. We can address them. We will, if there are really good questions that have come through from the chat, we'll actually call on the person to ask those formally. But I think with that, let me focus. The presentation's around 25 minutes, and then we'll have a 25-minute Q&A. So you'll have a little bit of time, folks, to actually get many of your specific questions, if they're not addressed in the presentation, answered. But really, we've all come here to hear about Dr. GAGANDEEP Kang, who's a doctor and a scientist. She spends most of her time in CMC Vellore, a microbiologist and a virologist, and clearly who has done a lot of research around viral infections in children, specifically around the testing of rotaviral vaccines. So this is a vaccine specialist. She's published in over 300 scientific papers, sits on multiple editorial boards globally, is the executive director of multiple institutions and advises multiple institutions, some that I've had the luxury and privilege of sharing time with. We both shared some time in Texas once upon a life, as I like to joke about it, ma'am. But I think you have perhaps used that time from Texas a lot more usefully and gainfully than I did. And a winner of multiple awards, she has received the Infosys Life Science Award, as well as known as the India's vaccine godmother, to name a few. The first Indian lady to be elected to the Fellow of the Royal Society, but we hope setting the platform for many other good people to start. And I think more importantly and more recently, you have co-authored a really outstanding publication on India's fight against the COVID pandemic with some great co-authors. So I think that's the platform. We have a real expert in the field. We have somebody who I think can ask and be asked some of the tougher questions. And we hope that this goes deeper, because as many of you are owners or are clinicians, we expect that you have some queries around the vaccine reality of today. And our hope is over the next 25 minutes of presentation, many of those questions. So Dr. Kang, the stage is all yours, please.
Thank you very much for the introduction, Sameer. I'm just going to share my screen. And I intend to provide an update on COVID-19 vaccines, but I think the greatest learning for all of us comes from the question and answer session. So it'll be great if we can get to that really quickly. So I'll keep the slides and the discussion around the slides just to set the stage for what I hope will be much more an interactive discussion. So when we evaluate vaccines, we take them through preclinical testing, then we take them through phase 1, 2, and 3, which is safety and deciding on the dosage, and then immunogenicity, and then efficacy, and finally vaccines are approved for use.
Please, guys, if you're not talking and you're not.
Mehta check kotte vela panchavatile try panna illa naanga thiruppi vena nanu ena already oxygen illa high ah irundhuchina konja idu dhaan pandranga kettu solren neenga avangaloda number idu vandu
I think Dr. Premlata is really struggling.
Name number meeting iruken
So where we are today is that we have 7 vaccines that are in limited use. Most of these are in Russia and in China. And then we have emergency use authorization for 8 vaccines. The first vaccine that is going for a full licensure is the Pfizer vaccine, and that's going to be considered by the FDA in the US. There are so far only 4 vaccines that have been abandoned after coming into human testing. And anybody who works on vaccines will tell you that this is an absolutely incredible strike rate, because usually once a vaccine comes into humans, you expect somewhere between a 1 in 10 or a 1 in 8 chance of the vaccine succeeding. And here we have 15 successful vaccines as against 4 abandoned vaccines so far. So the platforms that we have, at least a couple of those are not platforms that are widely used. We've never had mRNA vaccines before. And now we have 2 and potentially 2 more on the way, including one in India. we potentially have Zydus Cadilla's vaccine, which might wind up being the first vaccine to be licensed for humans. The results from Zydus Cadila are expected anytime in the next few weeks. And then we have vectored vaccines, and those, the first vectored vaccines only were licensed in 2019, and that was the vaccine for Ebola. Now we have the AstraZeneca vaccine, the Johnson & Johnson vaccine, the Gamaleya vaccine, and China's CanSinoBio, also an adenovirus vectored vaccine. The older platforms, which are shown on the top of this slide, include live attenuated and inactivated vaccines. So far, we don't have a live attenuated vaccine, but we have multiple inactivated vaccines. And we also have a vaccine that can be called either a virus-like particle or a recombinant DNA vaccine, which is the vaccine that is made by Novovax. That vaccine has phase 3 efficacy data, but is not yet being used because it has not gone for a regulatory submission as yet. Now, a year ago, almost exactly 1 year ago, I was on a committee with WHO, which was asked to define what should a vaccine look like? What should we expect from a vaccine? And our framing at that time was really about influenza. We thought this is going to be like a flu vaccine, which means that the vaccine will be very difficult to make, and to get a high efficacy will be very challenging. So we set the bar as the vaccine must have at least 50% efficacy. We should use that vaccine with not more than 2 doses. It should be a vaccine that is usable in adults and give protection for at least 6 months. We now know that practically every vaccine that we have with efficacy data is doing better than that. So shown on this slide is a list of all the vaccines that have actually gone through safety testing, immunogenicity testing, and have clinical efficacy data. So we really know that these are the vaccines that work. And as you can see, there are 4 different platforms: mRNA, adenovirus vectored, inactivated, and subunit vaccine. So 4 platforms on which all these vaccines are made. Now, we've been through an interesting exercise because the world knew that there would be a shortage of vaccines. So the question was, how do you decide who gets vaccine first? And WHO consulted with a lot of countries around the world and came up with a framework that said that the goals of the vaccination program are to prevent deaths, so protect public health first, which means that you first make sure that people can get healthcare, and you do that by immunizing healthcare workers and social workers. Then to prevent deaths, you go into high-risk adults, and after that, you can look at other priority populations. So high-risk adults are usually those who are elderly and those with comorbidities, and it was anticipated that the amount of vaccine that would be required to cover the first 2 categories, health and social care workers and high-risk adults, would be a total of about 20% of any country's population. So this is what was decided by WHO, recognizing that WHO would need to play a role in getting vaccine to poorer countries, whereas rich countries would have the freedom to give vaccine to their entire populations as they pleased. As it turned out, rich countries also followed the same prioritization framework, going after health and healthcare workers first, then high-risk adults. In the UK, with just an age descending strategy, and then other priority groups. So much so that now in the UK, in the US, they are immunizing all ages and have started to go down to 12 years. Whereas in the UK, they have now reached the age of 42-year-olds being able to book their appointments with dates for immunization sometime in July. So they know it's coming and they'll know when they'll get the vaccine. Other parts of the world have been variable. We know the success story of many small countries in the Middle East and Israel doing a really good job of immunizing their populations. And now China has really picked up and is immunizing its population at a much, much faster rate than it was doing in the first few months of the year. Now, in India, the initial announcement that was made by the Mystery Committee, NEGVAC, was that they would immunize healthcare workers, essential workers, and individuals who were over 50 years of age. When the implementation actually started, we started with healthcare workers and then moved to essential workers, then to over 60s, then over 45s, and now to 18 and above in a very, very rapid loosening of permissions. We are at a stage now where we expect to be able to, or we are supposed to immunize 900 million people. And if you look at what's happening with the vaccination program, just over 10% of the population has received at least one dose of COVID-19 vaccine. And after reaching an average of about 3.5 million doses, we have now gotten, we are doing half of that on a daily basis for immunizing our populations. So moving from that to just cover each of the vaccines quickly. Among the available vaccines, we have a lot of inactivated vaccines. This is an older technology. It's very easy to make an inactivated vaccine because you have to just grow the virus, treat it with chemicals, and then see that there is no virus capable of replication left. The problem with making these vaccines is that they all have to be made in a high level of containment, what is called biosafety level 3 laboratories. And to manufacture things at BSL 3 can be quite challenging. So in India, we have the collaboration between Bharat Biotech and ICMR that has resulted in the Covaxin vaccine. Bharat Biotech started out manufacturing in its Hyderabad facility, and now it is working on a facility in Pune and another facility in Bangalore that have BSL-3s and is hoping to get them up and running quickly. So far, there is not production happening at those facilities. In addition, the government has also given licenses for the inactivated vaccine to multiple other companies. I'm a little doubtful about how long it will take those companies to get up and running, because as I mentioned, BSL-3 manufacturing can be quite challenging. And then, of course, there are a bunch of other vaccines that are being used in other parts of the world, mostly vaccines that have been made in China, but there are also inactivated vaccines in Russia and in Kazakhstan that are in development. Protein subunit vaccines, this is the Novovax vaccine. This is variably described as a virus-like particle because it self-assembles into that pretty flower-like shape, which consists of the spike protein aggregating together, or you can see it in the form of protein subunits. The spike protein is made up actually of 3 pieces of protein that wrap around each other. And we use the spike protein because the spike is what binds to the ACE2 receptor. So if you make antibodies to the spike, those antibodies can block the virus from getting inside the host cells. The Novovax vaccine, it has been evaluated in clinical trials in the UK, the US, and in South Africa. And it has been shown to have 96% efficacy against the D614G, the kind of virus we saw last year, 86% against B117, and about 51% in people who don't have HIV in South Africa, which is the location where B1351 variant was circulating. And in their trial, 92% of the strains that they had in South Africa were actually B1351. So the vaccine's working at least 50% of the time, even against the variant found in South Africa. Now, originally, the Novavax was supposed to do a regulatory filing in the second quarter of 2021. It's just been announced that they're going to be delaying that to Q3 2021, which means that this vaccine is not going to be available to much of the world until much later in this year. And in India, this vaccine is going to be made as Covovax by Serum Institute of India. They've already started the production, but according to Serum, because of restrictions in supplies from the US, export restrictions that were imposed under the Defense Production Act by the Biden administration, they have not been able to ramp up as quickly as they had intended. There are other protein subunit vaccines which are also in development. We also have the amazing success story of mRNA vaccines. This is the first mRNA vaccine to be licensed for an infectious disease. And we have two of those, Moderna and Pfizer, and they seem to have incredibly high efficacy. So much so that you start to see protection from these vaccines 10 days after the first dose of the vaccine is given, and the protection appears to be very good. Because these vaccines are very easy to design, little harder to make, but easy to design, both Moderna and Pfizer have made vaccines that are against B1351. And they have also evaluated these vaccines in children. So they're doing a lot of very interesting studies with these vaccines, now giving boosters to people who got the vaccine at this time last year and seeing how much that affects the immune response. The good news is that the older version of the vaccine also induces a broadening of the immune response and gives better protection against the variants. So even if we do need booster doses of vaccines, we practically have them already. Now, this is real-world data on the use of mRNA vaccines. And what you're looking at here is how quickly do the lines separate among the unvaccinated in red and the vaccinated in blue. This is data from Israel on about over 1 million people, half unvaccinated and half vaccinated. And essentially what it shows is that the mRNA vaccines are protecting against infection, symptomatic disease, hospitalization, severe disease, and deaths. These vaccines are doing exactly what they are supposed to be doing, which is protecting against severe disease and death, but they are also protecting against infection, which gives us hope that if we can get enough people vaccinated, this can create a situation where we can go back to life that is closer to normal. It's not just about protection from disease, it's also about protection from infection and transmission, which are very important public health goals. Then we have viral vectored vaccines. We have the University of Oxford AstraZeneca Serum Institute of India vaccine, which is based on the chimp adenovirus. And this had trial data that was from the UK, Brazil, and South Africa. And this was a kind of messy study because there were doubts about, how much of a dose, how variable was the intervals between doses. But all of the information that came out in that messy study has actually been validated by data that came out of the US that showed that there was pretty good efficacy with this vaccine with a 4-week interval. So there is a discussion ongoing now whether this vaccine could also have been a single dose vaccine like the Janssen & Janssen vaccine, which is just the Ad26 vector, which has been shown to have 74% efficacy with a single dose in the US, 66% in Brazil, where there was a variant circulating, and 52% in South Africa, where again there was a variant circulating. Now, even though these numbers appear lower than the numbers we saw for the Pfizer and the Moderna vaccines, in the Janssen & Janssen vaccine, the protection against severe disease and mortality was practically 100% as well. So all of the vaccines do seem to be working and working very well. Sometimes the variability that you see in the numbers depends on how the clinical trial defined the primary outcome of the study. Now, this Janssen & Janssen vaccine is going to be made in India by biological teams, and it will be supported by the Quad initiative. So there is now real-world data from the AstraZeneca vaccine from Scotland and from the UK, showing that a single dose of vaccine protects really well against severe disease and hospitalizations and mortality, and that a dosing interval, a longer dosing interval is fine. More recently, we've just had data that shows that even a single dose protects against transmission within households. You expect households to be a high transmission setting, and the AstraZeneca vaccine with a single dose cuts household transmission by half. So these vaccines really are working, and that's why much of the Western world is beginning to get back to what might be closer to normal. So these are the data from the AstraZeneca vaccine with the 12-week dosing interval, showing that even in the elderly, you see a reduction in severe disease and death by 80% with one dose of the AstraZeneca vaccine. And that is the basis for the longer interval that we have proposed. And that one dose of AstraZeneca vaccine after 2 weeks decreases household spread by half. So AstraZeneca and Pfizer vaccine seem to be performing very similarly in real life, even though the numbers looked very different in the clinical trials. Now, coming to safety issues with these vaccines, with Moderna and with Pfizer, we know that anaphylaxis happens. This is, both of these vaccines are very inflammatory. And one of the issues with RNA vaccines, why we couldn't make them earlier, was because RNA is a very inflammatory molecule. But we see anaphylaxis at low rates. And with the AstraZeneca and the Johnson & Johnson vaccine, we are seeing a condition called thrombosis with thrombocytopenia syndrome, which is approximately 1 in 100,000. But the risk is somewhat higher in younger women. So that's why the AstraZeneca vaccine has been advised to be replaced by some other vaccines in women less than 40 years of age. They've extended it to include men as well, just to make it programmatically simpler. But the risk is more in younger women. And the risk-benefit calculation for the AstraZeneca vaccine really means that you use this vaccine less when you have very little disease around you. As soon as the rate of disease around you goes up, then the benefit of the AstraZeneca vaccine increases greatly and the recommendations change. So given the status in India today, the benefit of the AstraZeneca vaccine very, very far outweighs any small risk of thrombosis with thrombocytopenia. Now, in India, we have the National AEFI Committee, which has been doing causality assessment, and it classifies the cases. If you have A1, A2, that usually means that this may be something that is related to the product or the quality of the vaccine. And here is an analysis where with Covishield and Covaxin, you can see that there is reactions that were considered to be potentially vaccine product related reactions. So both were features of anaphylaxis with these two vaccines. Now in India, the current choice of vaccines is Covishield, Covaxin, and Sputnik V. They have different prices and somewhat different dosing schedules, different storage conditions as well. So far, 90% of what has been used in India is Covishield, 10% is Covaxin, and Covishield has the advantage of having lots of data from other parts of the world, and Covaxin is planning a study of 5,000 people in Brazil. Sputnik V has been tried in Russia and in Eastern Europe, and a small trial similar to the Covishield trial was done in India. The efficacy of Covishield varies, but as I showed you, the real life performance of the AstraZeneca vaccine seems to be better than the efficacy that was seen in the clinical trials. For Covaxin, we have interim efficacy data, and where safety is concerned, we don't have safety data for Covaxin and Sputnik, but this may also be a feature of how many people the vaccine has been used in. Whereas with Covishield and the Johnson vaccine, we see the TTS incidence. So coming quickly to variants, 21A is B1617, 1, 2, and 3. And you can see that it has begun to be described really only from March of 2020. We actually know that there was some of it around as early as October last year, but in India, we were doing very little sequencing. So it doesn't show up as sequences that have been made available in global databases. Now, with mutations, it's always, does a mutation really matter or not? I think one thing that's very important to understand is that there are lots and lots and lots of variants. Every time viruses replicate, a variant will emerge, practically every time. But most variants don't matter. So this is a tile diagram, and shown in the big block of gray is B117. And this is just to illustrate that B117 was a variant that grew and became a variant of concern, whereas the bulk of variants that we saw actually amounted to nothing and did not cause a lot of infections. So what can happen with variants, and why should we worry? Variants can be more transmissible. And we've seen that with B117. We are seeing that with B1617. What you can see here in the blue and red graph is the data from the UK showing what is happening to B16172. You can see that it is beginning to replace the dominant strain in the UK, which was the B117 or the Kent strain. So the Kent strain was more transmissible than the older versions that were circulating in the UK, about 50% more transmissible, and it took over. And now B1617 is in the UK, and you can see that it is climbing very, very rapidly. So this is a very transmissible virus. Viruses can also evolve to escape the immune response, and this has been seen to variable extents with B1351 and P1. Both of these have E484K mutations, whereas the Indian variant has E484Q, which seems to be somewhat less consequential than what has been seen with B1351 and P1. Now, the vaccine trials have shown lower efficacy when you have immune escape, but it's not that severe disease is not being prevented. Both Novavax and Janssen gave at least 50% protection. With AstraZeneca, the trial size was very small, and it wasn't clear that severe disease was not being prevented. The variant of concern, as labeled by WHO, has 3 sublineages. Of these, B1617.1 actually has a reduction in about 7 times its ability to be neutralized by anti-sera. But fortunately for us, the more transmissible one, which is B1617.2, has an antibody reduction of only about half, and we usually have enough of a cushion of antibodies to be able to handle a virus where the effect, the amount of antibody required is double. There are lots of myths about vaccines. I'm not going to walk you through all of these because I'm sure you've heard all of them and we can discuss those. But I'd like to tell you about just one myth and what the reality really is. And this myth is about vaccination and infertility. This came from 2 people, one a scientist from Pfizer, Dr. Mike Yeadon, and a former member of the European Parliament, Dr. Wolfgang Wodardt. They asked the European Medicines Agency to delay the study or approval of the Pfizer vaccine. And what they said was that there's a protein called Synctin-1, which is in the placenta, and it's very similar to the coronavirus spike protein. So with the vaccine, when antibodies are made to the spike protein, those antibodies will also be directed against Synctin-1, and the placenta will be attacked and women will not be able to have babies. Now, the important thing to remember is that the spike protein is in the virus itself. So if this hypothesis was working, then you should be seeing exactly the same thing in infected people, not just in vaccinated people. But just to give you an illustration of the data that they were basing their assumption on, here is an alignment of the residues of the spike protein and of Synctin-1. The spike protein has 1,273 residues, and the Synctin-1 protein has 538 residues. There is an alignment of a string of 4 residues. That's it. That's what they were basing their argument on. If you do the same thing, looking at Synctin-1 with the spike protein with a couple of other human proteins that are expressed not in pregnancy, but in normal life, like hemoglobin or collagen, actually you find that hemoglobin is more similar to the spike protein because it has 2 sections where you get an alignment of 4 and 5 residues. Whereas in collagen also, you see an alignment that is similar for at least 4 residues. So if you look at it that way, practically if you make the residue string that you are looking at really, really short, any protein you look at will be similar to any other protein. So this kind of statement is not actually based in fact, and it's important for us to understand that. There are sites that are available that allow us to check whether these facts are true or not. And at Yale, there are scientists who've been looking at the impact of the maternal antiviral response on fetal development. And they evaluated serum from women with COVID-19 and found that there was no reaction between antibodies for the coronavirus and the Synctin-1 protein. So I hope that that has been put to rest. Unfortunately, it continues to be something that is hyped. And as late as 2 weeks ago, there was a report about a woman who had a preterm birth, and they claimed it was because she had been vaccinated. And that is absolutely not likely to be the case. In fact, it's been shown that when you are infected with COVID-19, you have a much higher risk of preterm birth than if you are vaccinated. So just to end, what does the future hold for us for vaccines? Vaccines are working, they're working brilliantly. We just need to get them out as quickly as we can. In case variants come, we made vaccines the first time around in 10 months, and we absolutely can do it in less. We are now thinking, is 6 months feasible? And with the right regulatory pathways, we think it is. So we need to get people vaccinated. Unfortunately, we are in a situation in India where we just don't have enough vaccine. So we have to continue with non-pharmaceutical interventions, because the best way to prevent variants is to prevent the virus from replicating. If it's not replicating, it can't be mutating. And if we want to protect ourselves and protect the world, we need to make sure that we suppress replication and protect people. So thank you very much for your attention.
Thanks a lot, Dr. Gagandeep. I think this was really useful. I remember, if I rewind the clock to a couple of months ago, we were thinking that vaccine effectiveness was, I take a shot and it keeps me safe. It looked at efficacy was measured in simple stuff like transmissibility and how deep the intensity of the virus would hit a person. Today, if we look at it, it's things like viral strain protection. It's the longevity of those antibodies. It's the number of doses. It's the population specificity. It's the safety around adverse reactions. It's the cost of production, transportation, storage, and in many cases now deployment. So the vaccine science, I think, to a lot of the common man, as well as people who help manage hospitals or clinicians, has come to life in a manner that perhaps none of us really had imagined 14 to 18 months before. But those were words, and these are, I guess, criteria for effectiveness around efficacy and immunogenicity that are popular. Are these the right kind of criteria to think about when you're evaluating vaccines?
Lots of things. I mean, you can make a vaccine, but ultimately it is vaccination that protects people and populations. So you do have to make sure that you have the right storage conditions, that you're not making the delivery something that is overly challenging. It should be easy to give vaccines. So the ideal vaccine might be a pill that you pop every 6 months. And there are people who are working on oral vaccines that are like that. But at the moment, what we have is injectable vaccines. And that's what we'll have to work with for this wave of the pandemic.
So just to build on something you mentioned, because we've had some questions, oral and nasal have been talked a little bit about. What are the fundamental differences between an oral and nasal? Or are they just different types of risks, easier to administer when you look at those two versus what's currently there?
So Sometimes you want to give a vaccine that is given by the route of natural infection. So for example, for polio, when you give an oral vaccine, you get local or mucosal immunity, which you don't get with an injectable vaccine. So if you want to prevent infection in a community, you would prefer to give that oral vaccine rather than the injectable vaccine, which protects the person but doesn't protect the community. Nasal spray vaccines have been tried for a while for influenza, for measles. The only one that we have licensed is an influenza vaccine, and it doesn't work as well as an injectable vaccine. So I know there is a lot of hype right now about nasal spray vaccines, but I think we need to wait and see how they perform.
Okay. I'm beginning to look at some of the questions. We have a great question from both Dr. Thangavelu, Dr. Sarvanakumar, and a few others on the boosters. Clearly, we never spoke, 9 months or 12 months ago about the fact that this would be an annual health recipe requirement. But now we're beginning to hear that there is something called an annual booster. Are there any booster studies that you are familiar with, or you think that it's still too early and they'll start coming through?
So I mentioned that both Moderna and Pfizer are trying boosters on their phase 1 participants. And the results from the boosters are showing a broadening of the immune response, which means that they will protect better against the variant viruses. Is a booster really needed annually? We don't know that yet because we don't know the duration of protection by these vaccines. Given how well the vaccines are working, I think if we can get replication and the emergence of variants under control, we may not need annual vaccination. It may be every few years, or it may be a series that you take right now, and then it settles after a while. We'll just have to wait and see about that.
So let me try and merge a few different questions together, and I hope I don't make a hodgepodge. Which vaccine would you recommend for pregnant women in the universe of vaccines available, assuming you could travel anywhere in the world?
Any vaccine. Any vaccine, because currently all of the vaccines that we have are equivalent to non-replicating vaccines. So there is no reason why pregnant or lactating women should not be getting these vaccines. There are data now from the US on the Moderna and Pfizer vaccines, where they have been given to 35,000 women that have outcomes from their pregnancies, and there are no issues with these vaccines. We also don't expect an issue with any of the other vaccines because none of them are replicated.
Okay, and for children?
For children, I don't see the vaccines being any different from the ones that we have. It's just that they haven't been evaluated in children. So what we might want to do is adjust the dosing schedule a bit or adjust the dose a bit. But at the moment, there seems to be no reason why children cannot get these same vaccines, except that they are not yet, other than Pfizer and Moderna, they have not yet been reviewed by regulators.
Interesting. So it's a lot about data availability and the reality there. A very different type of question. I think it's a pretty good one in terms of which would be the lab test that's readily available that would tell or at least showcase the recipient immunity. Is there something that you recommend, or take the vaccine and hope?
Everybody I know is going out and getting their antibodies tested, complete and utter waste. So we don't have a correlate of protection at this time. Once we have one, that's the test to use, but it's likely to be neutralizing antibodies, but which test and what level of neutralizing antibodies, we don't know yet, and we will probably not know for another 3 to 6 months.
And I guess that's similar to, we clearly don't have sufficient data around complications of any of the vaccine mid to long term, because that just hasn't that's not there yet, right? Are there,
let's be very clear. Most of the side effects you have from vaccines will manifest themselves in a couple of months, mostly a shorter time frame than that. That's why the FDA recommends that when you do safety data, you should monitor for at least 2 months. Most other trials, if they really think that there is something going on, might monitor for 6 months, but you don't expect a vaccine-related side effect to appear 2 years or 3 years after you've received the vaccine.
So I have two other questions. The first is, and I think this might just be the reality of numbers, we're seeing a few people obviously transitioning or dying after they've taken 2 doses of nearly all the available global vaccines. And clearly the percentages must be very, very, very small micro fractions. But because these are being hyped on social media, I think a big chunk of our populations are getting deeply concerned. Should we even care about this? I mean, obviously as scientists, there is a reality, and as the vaccine business there is, but as clinicians or hospital owners work exactly the other way to defend against. What are your views on this?
I think communicating vaccines and the role that vaccines play in protecting us is very important, particularly for people who are immunocompromised or elderly. They must understand that their immune responses are not going to be as good as other people, and they need to make sure that they protect themselves. So if you are on chemotherapy, if you are on long-term steroids, if you've had a transplant, you should expect that your protection will be less good than other people who don't have these conditions. And therefore, you need to make sure that you continue to protect yourself until the disease around you decreases to a very low level. Remember that you can only get infected if people around you are infected, and as long as infection around you is high, you are at risk. Once infection around you declines, then it becomes safer for a person who has only partial protection. But even if you're completely healthy and you do well, with most respiratory pathogens, most mucosal pathogens, a lot of immunity depends on the infectious dose. So if you get a really whacking dose of the virus, it may be that you would have been protected against a smaller dose of the virus, but with a large dose, you still get infected and get sick. So hopefully, vaccines protect against severe disease and death, but it's not perfect protection. Data from the US shows that of 75 million doses of Pfizer and Mrna vaccines that have been given, 75 people have died after receiving both doses of the vaccine. So it's a 1 in a million chance, but it's still 75 people who died. About 4 times that number got hospitalized and had severe disease. So it's not like vaccines are perfect protection. Very good protection, but not perfect.
A scientific question from our Dr. Nedinchalian, which is, what is the mechanism of immune escape? I hope you're able to follow that.
Yeah. So the mechanism of immune escape, well, if you go back to school and college, we used to think of antibody-antigen interactions as locks and keys, right? So you have to have a certain shape of key to fit that particular lock. If the lock changes, then the key won't fit it anymore. And that's essentially what happens with proteins. So protein that changes its shape will no longer be bound by the antibody that was made to an older version of the protein. That's immune escape.
So I think I've got a bunch of questions and I have to acknowledge a lot of clear scientific. I think we have one from Dr. Aditi that I really don't understand. So in the case of Bell's palsy, following the first dose of vaccine, is the second dose contradicted? And I guess it's linked to another one. If you've had an adverse reaction to the first vaccine, how should you think about the second one?
So there's been the cases of Bell's palsy that were reported during the clinical trial have not manifested in any numbers once the vaccine started to be used in multiples of millions of doses. So that may have been just a chance finding. Right now, it looks like rates of Bell's palsy are not increased by vaccination. Currently, there are a lot of questions I can see about contraindications. There are no contraindications to taking these vaccines except an allergic or an anaphylactic reaction to the first dose of the vaccine, which means that you don't use a second dose of the same product. So that is the only contraindication to taking the vaccine.
So I think thanks a lot, Dr. Kang. I should end with something that's a little bit more uplifting. Nearly everyone I suspect on this video has probably, or this live channel has probably taken 2 vaccines. If they haven't, advice 1 is take the vaccine. I think that's been repeated a few times. But advice 2, what should you do once you've taken the 2 vaccines beyond obviously limit the number of social interactions or avoid them unless completely necessary. And unfortunately, most of the folks on this call are healthcare workers, professionals, or leaders. How should we think about it differently, Dr. Kang?
So right now for healthcare workers, because there is so much disease around, we still, even if we are vaccinated, we need to make sure that we stay protected. And for that, the quality of the PPE is very, very important. Once disease around us comes down, then it becomes possible for people who are protected by vaccination to think about less investment in non-pharmaceutical interventions. Right now is not the time for that. Vaccines work very well. If we can get enough vaccines to enough people, we will begin to see the change.
Thank you. I think we're trying our best in AHPI. I think so far it's close to 10 million doses have been earmarked for small and medium-sized hospitals. That will be part of a problem to help at least vaccinate more people. But it's only a drop in the ocean, but hopefully many drops make it an ocean. So I think with that, Dr. Kang, thanks a lot. This was a wonderfully scientific as well as educational session for folks like me who perhaps don't spend as much of the day knee deep in this. I think as with all things positive, we should look at the best, and that is compared to 12 and 14 months ago, we're much more educated, we're much better protected, and we really know the people that love us because they're the ones who check in on us every day and regularly. So I think with that, let me thank you, Dr. Kang and Gagandeep. And Gayatri, over to you. Thank you.
Thank you, Sam. Thank you, Dr. Gagandeep, for this very vivid illustration with a very simple presentation to make all of us understand how all the vaccines work, their plus and the minuses. So much of work is going, much is happening around. We are fighting a disease that is an unseen enemy, and people like you have made our lives so hopeful and better. Thank you so much for this lovely conversation also, Sam. I think good questions elicit the best answers from people. So this conversation was so helpful, and audience, you've been really good and really wonderful to see so many questions pop up in the chat box. So it is such a lovely time we had. Thank you so much for being so illuminative in your talk as well. So thank you very much. Before we close the program, I would like to just go back to my presentation to show a few numbers for the benefit of people who would like to join AHPI. I'm sure a lot of healthcare providers are watching this, a lot of hospital owners, small hospitals, large hospitals. in and around Tamil Nadu, entire India watching this. For people who may not know about AHPI yet, please welcome to log in and download the app. AHPI Tamil Nadu app is already there. You can download and look into the information that's being provided. Apart from this, we have a lot of benefits for healthcare providers as such, if you can become members of AHPI. So from my presentation, the last 2 slides, please, so that I can give them the numbers, the contact numbers for people who are healthcare providers can actually get in touch with us to enroll as members of AHPI. So this is our structure. We have all the members listed here. We have very specific roles. Very interesting, very important information is being shared in our groups, and very useful stuff is being negotiated with the government on a daily basis through AHPI. And whatever is being done is not just benefiting the members of AHPI, is also being passed on to the entire world of doctors and the communities that treat people. So these are the roles. And next slide, please. So these are the reasons why you should join AHPI. Download the AHPI app, please. And then if you are a person who requires a personal growth in the healthcare industry, if you want your hospital to be seen, if you want institutional support and accreditation, please join AHPI. Update yourself on the latest technology that's happening around. Legal help or any verification of costing, if you want to be on par with the current costing that's happening around of the same sized hospitals as you are, we will help you out. If you train your manpower, we have excellent training programs. Please enroll with AHPI for this. We help in accreditation. We have a lot of association with institutions like CAHO, which are dealing with accredited hospitals and with accreditation bodies like NABH, JCI. And of course, finally, we make the best friends out here. They're all friends for life. They're just not professional friends, but they become quite close to the families as well. So if you are a person who likes to socially network as well, so please join AHPI. This, of course, as I said in the beginning of the program, is a membership drive, and it's a member connect program. So I thank each and every member of AHPI for giving us this platform and support that we are able to conduct programs like this for a greater good. I especially thank our speakers, all the people from the center who have been great support, Dr. Alex, Dr. Giridhar Gyani, and of course, Shikhar, who has helped us put up these presentations together. I thank you very much. And of course, how can I not thank our partners, CII, Emergency Medicine Society of India, and Annie as well, for giving us support in this program. I thank our moderator, Mr. SAMEER Mehta, for being such a great, wonderful moderator of questions and compiling the whole show together. The key contacts, now you can take a picture of this particular slide so that if you want to become members of AHPI, please contact these numbers based upon the geographical position of your hospital. We pretty much support you in small as well as big things. Anything concerned with hospitals and healthcare, please, you can contact us. Thank you so much. With this, I would like to close the program. And I thank very much the support system from Mehta Hospital for providing us the Facebook link and also making this program a great success. So wish you all better times. I hope to meet you soon.
Thank you, Gayatri. I think also we should thank AHPI and IBAR, who I think were strong partners of ours through this program. Thank you.
Very much.